the soil living organisms are very much depleted. Some scientists say that we have uh, topsoil where we actually grow our food for the next 60 years. Well, they said it 10 years ago, so we can say that for half a decade we still have some agricultural soils, but with the rate that they are decaying today, we might not have enough soil
Excuse me,
50
Felicia.
years. Uh-huh, go for it.
Are we going without the PowerPoint?
You don't see the PowerPoint?
No, I
Yeah,
don't.
it disappeared. I don't know.
Oh, it disappeared. Oh my God. Okay, I'm really sorry. I'm gonna do it again. You see it now?
Now it's okay.
Okay. Thank you so much for letting me know. I really appreciate, and anytime it disappears, please let me know, because I have some really pretty images here that I would like to show you. So I really appreciate. Thank you so much. So um yeah, so soil degradation. And really, these days, when I'm traveling across the country at winter time, you don't really see much cover on most of the soils. And the wind blows away the soil, precipitation, rainwater washes away the soil, which is a huge issue. Erosion is a huge issue in terms of soil degradation. Um, and here is a map of the state of our soils worldwide. And I'm going to get a laser pointer. So basically, with red, you can see very degraded soils. You probably are able to find your own country here and see whether it's very degraded or degraded. As you can see, most of our temperate and Mediterranean regions and tropical regions are very, very much degraded. Really, we can only find stable soils in Canada, in Alaska, and in Siberia, and a few patches here and there. Um, which is a huge concern, and we really need to do something about this. And as Franklin Roosevelt, who is an ex-president of the United States back in the time, he says that a nation who destroys its soil destroys itself. And we have seen this many times in the history, that actually, if you think about the old Mesopotamia, the Egyptian cultures, and so on, so many cultures disappeared. And partially it is because they were not able to grow food anymore, because they degraded their soils. But yeah, so what is soil really? When you take a handful of soil, you just reach down and take a handful of soil, you would think that for the most part it is a solid material. But in reality, in a healthy soil, that material is maximum 50% solid, the mineral part of the soil, the sands, the silt, and clay that you can actually feel in your hands. The rest of it is pore space, smaller or larger spaces which can be filled with air or water, or organic matter is also what is in that pore space. And really, that I show here 5% organic matter, but in our agricultural soils, depending on the soil type, that can be even as low as 1% or less. In many places in Hungary, and probably in your countries as well, you have barely 1 or 2% organic matter in agricultural soils, which is a big issue, because this is where actually everything happens that is important in terms of soil productivity, in terms of the food that we are able to eat. So this organic matter is actually composed of various type of stuff that come from living organisms. So about 5% of that tiny fraction is living creatures, and I'm going to show you some living organisms that actually inhabit soils. And then there is fresh residue, which means that those organisms, mostly plants and other animals, when they die, or when leaves fall, or when roots die, you get this fresh residue component that starts to decompose, and they call it the active fraction. And then there is also a stabilized fraction, which some people call humus. Actually, I call humus everything that is in the yellow, the purple, and the green. So there is no scientific consensus about what humus is, and I'm going to talk to you a little bit about this one too. But let's have a look at the carbon cycle. This image is actually simpler than you would think, and let's focus on soils here. You see, these numbers all are in pentagrams. Okay, so we have a carbonate rock base that has a really a lot of carbon, and there is a little bit of exchange, through fossil fuels, and a little bit from the oceans goes back to these rocks. The ocean holds a huge part of this carbon. And why we are talking about the carbon cycle, as I said, the organic part of this soil is actually mostly carbon. Carbon is the building block of life. But I'm sure you have learned about it before. So let's focus a little bit on soils. Okay. Soil has a huge reservoir of carbon that is actually able to move out of the soil. Okay, and as we are talking about climate change, a lot of this carbon is going to the atmosphere from the soils. So basically, vegetation through photosynthesis is taking up a good chunk of carbon. It says 110 pentagrams. Obviously, this is an estimate. And then 50 of that goes back to the atmosphere as plants decompose, and probably they also exhale some carbon dioxide and so on. And 60%, 60 pentagrams, I'm sorry, I'm talking about percents, but this is not percent. 60 pentagrams is staying in the soil. And but the soil also exhales some carbon dioxide, and you can see that there is 2 pentagrams more carbon exhaled, moved from the soil than what is actually getting into the soil, actually 2.5, right? So basically there is more carbon being lost from the soil than what is getting into the soil. And this is a huge problem, and this is actually the reason for organic matter depletion in our soils, which is well visible in this carbon cycle. And then, of course, you also see fossil fuel burning that we use for our industry, for our transportation, and of course, for a good part in agriculture, and that is also getting into the atmosphere. And there is nothing that actually captures that carbon back into our systems. Okay, so this is the carbon cycle. This image comes from this textbook, which if you ever get interested in soils, I highly recommend getting this book because it has a ton of really valuable information about basic soil science with a good foundation also in sustainable agriculture. Okay, so probably this is going to be the most complex image in this whole lecture. So basically, I don't want to get too deep into this, and if you have questions, please feel free to interrupt. This can be also a discussion. But what was a huge revelation for me, and I think it's a very important concept, that back in the time when 150 200 years ago, when the first chemical experiments showed up, we wanted to know how nature works. And of course, by today, we realize that nature is much more complex than we have ever imagined. But there is this fundamental desire to be able to kind of like define everything. So, of course, for over a century, scientists were trying to define what is humus and write down a molecular structure for humus. This is a humic acid molecular structure. So one of my first revelation was that there in the world there is no such uh, material present. And of course, in every humic acid molecule is different. There is no two humic acid molecules alike. And it would even be extremely hard to write down any sort of chemical composition for that. It has different functional groups that makes humus a very active material that is able to hold on to nutrients and so on and so forth. And also, science said that there is a strange chemical process, physical chemical process in soil that makes decomposing organic matter into this structure. And then when it reaches this complex structure, it is able to be persistent in soil. Now, the reality is that there is no such, most likely, there is no such miraculous humus molecule that is able to withhold to degradation. You will see later that it's a much simple process. And there is these two scientists who actually created the soil continuum model, which is response to the old humus theory. And they actually believe, and this caused a lot of stir in the scientific community. You know, when there is something that most scientists believed for decades, if not centuries, and then some people come and just turn it over and say that you are wrong, that causes a lot of anger, of course. But I tend to go with this model. So basically, how it looks, you know, how this organic matter functions in the soil, you know, that those living beings that die and then start to decompose and start to feed this system again. So first we have the plant and animal residues. They contain large organic molecules, carbohydrates, proteins, lignins, lipids, and pyrogenic compounds. And these, as soon as, you know, for example, a bacteria dies, it is able to form into aggregates, and also it's able to get attached to mineral surfaces. Okay, but there is life happening, and there is somebody who's for whom these plant and animal residues become food. So as they eat them, chew them, they become smaller. Some of that is pooped out, and these become large biopolymers. And again, these are able to form aggregates, and they are able to get absorbed to mineral surfaces. And the smaller the molecule is, it's more and more able to form aggregates, and more able to get attached to mineral surfaces. Okay, and then there is this small biopolymer. They become even smaller, and then eventually they become monomers. Monomers are, for example, like a sugar molecule or some sort of a protein molecule. They are no longer chains. They are just simple organic molecules. And then the end of the story is carbon dioxide. And then that carbon dioxide goes back into the air, and the plants take it up again. And what we would like to do in agriculture and generally in preserving soil health, we would like to have as much as possible from these molecules, these materials present in the soil. And that is not true that there is a miracle humic substance that is not able to decompose, and that's why it stays in the soil. There is a different reason, and the reason is aggregation and mineral absorption to mineral surfaces. I don't know if this is clear or not, but feel free to ask any questions. This is way much more simple than it may sound first. Okay, so let's talk a little bit about microbial necromass. Necromass is actually microbes that die and their dead bodies are there, and they are very, very important for the purpose of soil life. And there is this beautiful sentence which says that microbes are the eye of the needle through which all the organic materials must pass, which means that in this process that I explained, everything is happening through the action of microbes. And however simple it sounds, this is fairly a new understanding, because we have been thinking before that actually it's mostly chemical and physical processes happening in the soil. And actually it's the biology that runs the entire show. And everything that it has ever lived has been chewed by microbes, and that's what actually, like, every molecule, everything has been gone through the digestive system of microbes, and that's what cycles every material that is related to living materials, if you know what I mean. So basically, the dead microbial mass consists mainly of bacteria, fungi, because they are the dominant component of the long-lasting soil organic carbon. So the point that I would like to make here is that, as opposed to the previous views, and I'm not sure, I couldn't or forgot to ask you whether you have studied soil science or not. But the previous view was that humus has been created by this miraculous chemical process that makes these organic materials stable. And that is probably wrong. And in reality, what is persistent to decay is bacterial and fungal cell walls. Like, for example, fungi contains chitin, chitin, this really hard material that for example, insects have on their outer body. So fungi has chitin too, and that is very persistent to decomposition. So one thing is that these bacterial and fungal cell walls are persistent to decomposition. And that's how, if we have a lot of bacteria and a lot of fungi that actually also happen to die, that means that that is a way to increase soil organic carbon, which can also be very important in terms of climate change, because we know the capacity of the soil to actually store these materials. Actually, yeah, I believe that pretty much the only way to, if we ever going to be able to slow down climate change, that's going to happen through soils and plants, and plant cover on as much surfaces as possible. Okay, so this is a really interesting study that was done by Austrian and I think German scientist Anja Miltner and her team, and this is just to actually underline what I've said before, that actually soil organic matter is its formation is actually through microbial biomass. And what they found, they were looking at organic patchy fragments of 200 to 500 nanometer size. That's basically 500 nanometer is about half a bacteria. One bacteria is about 1 micrometer, and 500 nanometers is about half a bacteria. And they were looking at soil samples through an electron microscope. These are electron micrographs of what they actually found. And they found that these empty cell walls of bacteria, they call it patchy fragments, are actually what make up most of our organic matter. Okay, and then there is another amazing finding, which is just so obvious if you think about it. And it says that organic matter turnover, which means that how fast organic matter is being moved back to the atmosphere, like how fast the organic pool of soil organic matter becomes CO2 essentially, and then it goes back again through plants into the soil. So this turnover, she says, Jennifer Dungate, that is actually governed by accessibility, not recalcitrance. So what I have said so far is that soil organic matter is decomposable. There is no miracle soil organic matter except some bacterial cell walls and fungal cell walls, which is pretty hard to decompose, but it's possible. So everything is decomposable. Forget about humus theory. Everything is decomposable. But if that is the case, what preserves it? How can we preserve organic matter in soil? She says, Jennifer Dungate, 1, occlusion within aggregates. As I said before, they are able to form aggregates. Aggregates are these, when you take a handful of soil and you start to crumble it in your fingers, these little crumbs. A healthy soil has about 1 centimeter crumbs. That's what's called aggregates. And there is different size aggregates. I probably have this in this presentation to show you. And then absorption to minerals. This is also something that I showed you already in the soil continuum model. And then substrate-driven biological rate limitation. This sounds pretty complex, but in reality, again, this is very simple. For example, substrate-driven limitation could mean that there is a bacteria who wants to eat something, but it cannot because there is, for example, not enough nitrogen to be able to digest that. So this means substrate-driven limitation. And so accessibility, it's really basically how microbes or their exoenzymes can reach organic matter. So here is something that a microbe would like to eat. And is it able to reach this food? Like, for example, if you would like to get from your study room into the kitchen and there is all these furnitures and everything put in the way, it takes you forever until you are actually able to get there. But you can move that furniture away and you can get a free access. But if it's locked for some reason, for example, you have a lot of water, that is flooded your house and you cannot swim, then you will not be able to reach that organic matter. Or if it's desiccated. Okay, so many of microbes, if there is not enough water, much of these microbes move in water films, but they also need oxygen. If there is not enough they also need water to move. So if there is no water, they won't be able to reach it. But if it's waterlogged, there is not enough oxygen, so they die. So basically, physical accessibility is what preserves organic matter or not. Okay, so basically whether they are able to reach that piece of organic matter or not. That's simple. It is as simple as it is. And inside an aggregate, it is harder to reach. And outside, if that particular organic matter is outside of an aggregate or on the surface of an aggregate, it's easier to reach. If it's if there is too much water, it's hard to reach. If there is not enough water, it's hard to reach. Absolutely simple physics. Okay. what happens when there is actually, like, if we plow the soil too much? As I showed you, this pore space consists where all these things happen, where there is water, where there is air, where there is living creatures that actually make all this organic matter turnover, nutrient availability, and so on happen. That is, at least in a healthy soil, at least 50% of that soil. But if that soil has been way too many times plowed, turned over, too much organic matter is exposed to oxygen, organic matter moves out into the atmosphere, and this entire soil structure collapses. And here you have this plow plane, which means that it becomes a very compacted layer. So basically, roots have very hard time to penetrate. Most roots are not able to get down here. So here it's very hard to access any organic matter, but also it's very hard for organic matter to actually accumulate. And in such a soil, basically the mineral component can be up to 75% with a very, very little amount of organic matter. And most of our agricultural soils become extremely compacted. And that is not making soil life possible to a degree that is actually necessary to maintain healthy soil conditions. And, you know, when you plow the soil, basically what happens, this entire soil structure is falling apart. And where would you choose to live? Imagine that every single year, when the soil is plowed, that structure that the microbes and the plants build together is being ruined. Every single day, every single year, we ruin the homes of these creatures, and they start to rebuild it. But because there is less and less of them, and the soil is more and more compacted, it is harder and harder and harder to create a home out of these piles of bricks, really. So imagine soil aggregates as if they were the homes, the beautiful castles, and the highways and the cities of these soil microorganisms. And there is different sizes of aggregates. They start from tiny, tiny little aggregations that are made out of mostly clay and bacteria. Bacteria usually surrounds a little piece of organic matter, and then that is being surrounded by soil particles. And this is mostly on the surfaces, okay? And bacteria is generates or creates sugar-like substances. They are called polysaccharides that glue them onto the surfaces, glue them onto mineral surfaces, and also glue them to organic matter surfaces. And then these tiny aggregates become microaggregates when they are bound together by mostly fungi. And also this becomes, so these are fungal filaments, and fungi also creates a glue. For example, there is one material that is widely researched, is called glomalin. So they excrete glomalin, and this glomalin sticks these particles, these microaggregates together. And this is the home base for the microfauna, for example, nematodes and protozoa, and so on. Those microbes that actually feed on bacteria and fungi. And then there is the macroaggregates. The macroaggregates are the ones that you see when you crumble this soil that you take into your hand. These 1 centimeter to 1 millimeter size aggregates. So basically inside these aggregates, there is a lot of fungi, and plant roots can penetrate these aggregates. And also these are the rhizosphere, the rhizos the rhizosphere aggregations. I don't know if you guys have ever pulled out a plant from the soil, and you might, I will show a picture of this, seen these Rastafarian roots. They look like Rastas, soil aggregates stuck onto plant roots. Those are very important. It's called the rhizosheets. I will talk about it a little bit later. Okay, so basically the most important message here is that we need to preserve these soil aggregates, and the only way we can do this is through the action of microorganisms and organic matter. And if we don't have that, we have this instead. There is nothing to hold soil particles together, so rainwater, runoff water, is able to just wash our soil, our topsoil, and even subsoils here away. This is an image of an aggregate and the pore space. So these are the mineral particles. I hope you see my pointer. And then in the pore space, you see many organisms. And these pore spaces are either filled with air or water. You see a plant root here, a mycorrhizal fungi that is scavenging nutrients for the plant in exchange for carbon. You see an amoeba here. There is this mite that thinking about, oh, there is this amoeba, I would like to catch it. That would be a nice lunch for me. There is some bacterial aggregates, bacterial colonies that this amoeba would like to feed on. You see a nematode that is seeing these tiny organisms, probably bacteria, maybe some protists. It's like, hey, I would like to reach you and eat you. And they are like, no, you are not going to reach us here. A ciliate here. So basically, the aggregates and the pore space is where all this life is happening. And of course, it's not an accident that so many people like to say that there is more life in a teaspoon of soil than people on the planet. So how does the soil food web look like? So everything that we talked about so far relates to the soil food web. Okay, plants photosynthesize. They have a root system. Along this root system live fungi and bacteria. And also the plants exude, I will talk about this again, some exudates, also mostly amino acids, sugars, etc. that feed the bacteria. So they make sure that bacteria is living along their roots. And then bacteria is eaten by mites and protozoa. These are ciliates, flagellates, ciliates, and amoeba is not pictured here. And then also nematodes. And when a nematode eats a bacteria, here in the root, next to the root, actually in the rhizosphere, when that nematode is actually excretes, takes a poop, that's what the plants are able to take up. And this is the reason why a forest or a native prairie doesn't need any sort of fertilizer, as opposed to our agricultural systems where the soil is degraded, where this living system is not alive. That's where we need to intervene with chemicals. But when we use chemicals, we degrade this system even more. So this is a vicious cycle, but luckily it's possible to turn it around. So basically, higher and higher organisms eat each other, and that makes nutrient available for plants and lower organisms. That's the whole point in this system. And we shall not forget that we are also part of this system. We as human beings are part of the soil food web. Yeah, okay, I talked about photosynthesis, you get the sunlight, this beautiful energy that makes the carbon dioxide from the atmosphere into solid material. Plants are able to fix this carbon. I think this is the largest miracle that is happening on this earth. And then there is a root system. Much of this carbon dioxide that is taken up by plants is pumped into the root system. And probably I have a nice picture here that actually 20 to 40% of this photosynthetic carbon that plants take up is pumped into the soil, into the rhizosphere. This drop of liquid you see here at this root tap as root exudate, which I mentioned before. These are from simple sugars to more complex materials and have many different functions. And you can, when you imagine a plant that is pumping out these root exudates, the plant is actually deciding what material to exude according to what the plant wants, what kind of nutrients it wants. That he is going to, or she, they are going to feed the right bacterial population that is able to provide a service for them. And when a larger predator comes and eats that bacteria, the plant is able to take up those nutrients. But there is also, of course, exchange between plants and fungi. So there is a various area of exchanges. And then another portion of these plant root exudates are there to provide, for example, protection for the plants. This is one way how plants are able to signal to each other or to the bacterial colonies around their roots and gain protection. They exude enzymes, they exude hormones, signaling molecules, and so on. They are way much more smart than we can even possibly imagine. Okay. And then, of course, they recruit bacteria for various purposes, as I said. And here is an electron micrograph of a rhizosphere colony. You see these cocci-shaped bacteria. This is a very predominant, and also the rod-shaped bacteria, the Bacillus species. And then you also see some fungal filaments. So on the micro level, there is a lot of things happening in the rhizosphere. And here in scientific terms, what I said before, basically, immobilization means when these creatures eat something, okay? Eat another microorganism or just eat free nutrients from the soil solution. And then mineralization is really when they excrete, when they poop, they urinate in their own way, of course. That's what mineralization is, and that's when plant nutrients become available. And the way they behave is really very similar to how how we behave, you know, when we use the toilet. For example, urine is mostly urea, which is a predominant fertilizer in agriculture, the very same composition as our urine. So that is able to fertilize plants. And of course manure as well, but there is many things that you need to do before application. Okay, so it's very simple. Who eats whom, who poops what? That's the story of life. Okay, I'm going to show you a few images of these microorganisms that I have taken through. I'm doing soil microbiology analysis for different clients, for growers and compost compost makers, companies, and individuals. And these images come mostly from composts that actually I got from different people. But the soil microorganisms are very similar to these. So in this first image, you see various types of amoebae. Amoebae in the soil and in the compost usually live in a silica shell. If you Google electron micrographs of testate amoebas, it's just a wonder. They are amazingly beautiful, and the structure of their silica shells is just magnificent. And then also there is this Archiella. This is a different type of testate amoeba. There is so many different kinds of amoeba that it's even hard to imagine. And then there is a naked amoeba. This is much more rare in soils. And then this image, you also see a fungal spore. All these tiny dots are bacteria. There is another amoeba, a testate amoeba. There is a mineral particle. There is decomposing organic matter containing gazillions of bacteria. And then here you see a bacterial feeding nematode. The way you are able to know that this is a bacterial feeding nematode, because there is a different kind of nematodes, okay? And some of them like to eat bacteria, some of them like to eat fungi, some of them like to eat other nematodes, and so on. So basically, the way you know that this is a bacterial-eating nematode is that actually one bacteria just rightly fits into their mouth. And there is other features that you can see to recognize them. And this is a mite. And most likely this mite is dead and being eaten by bacteria. And their skeletons and their shells are their parts that decompose the slowest. This is a video of nematode colony. In the middle of the picture, you see a organic matter patch and kind of like an organic matter aggregate from which many fungal hyphae are growing. These are breaking off as I was shaking the sample. And these nematodes are having a feast. There is something really, really yummy here. And most likely they are fungal feeding nematodes. This is 100 times magnification. So they are 100 times smaller than what we actually see in the image. And this is a fungal feeding nematode. And the way you can differentiate them is that they have a sphere in their mouth part that they use to puncture the fungal hyphae and suck out the contents of this hypha. This is a ciliate. Ciliates like to live in soils that have soils and composts that have a little bit too much water. So when you see a lot of ciliates, that means that that soil probably is waterlogged or a little bit more moist than it should be, because they like to live in a lot of water. And the next one is also likes to live in a lot of water. This is a rotifer, and they have these coronal cilia that they are able to make a current with and force water to flow into their digestive system. And from there, they are able to basically get the nutrients that are in bacteria and algae and so on. They are really funny, beautiful creatures. This is a fungal hyphae, probably a saprophytic fungi. We really like to see fungal hyphae that is pretty thick in diameter. These tiny black dots are bacteria that are in focus, and they are most likely about 1 micrometer thick. And usually we compare them to the fungal hyphae, and that's how we predict or estimate the fungal diameter. So this is at least 8 times as big as this little bacteria, but that means that this is probably about 8 micrometer thick, which is a pretty good size for a fungi. And we like to see fungi that has this beautiful brown color. Fungi is very, very sensitive to disturbance. So when we plow the soil, we are cutting these fungal hyphae apart, and they just gradually disappear from our soils. From a conventional agricultural soil, where most of our wheat and corn and everything is being grown, we hardly see any fungal hyphae. It's very sad because they are very important from nutrient transportation perspective, from organic matter decomposition perspective, from communication perspective, and so on. A mite, oh no, Bochan, I'm sorry, this is not a mite, this is a collembola. A springtail. These are really cute creatures. They like to have, they really like fungal dominated soils and composts. They like to eat fungi, and they have this interesting leg that is usually turned up here. And when they see some danger, they just suddenly let it out. And a springtail that is about like 0.5 millimeter big is able to jump 0.5 meter. So they are big jumpers. That's why they are called springtails. Mites, I already talked about mites. They like to shred organic matter, eat bacteria, eat, mostly organic matter and bacteria. And some of them are predatory and are able to eat larger creatures as well. And of course, earthworms. Earthworms are the centerpiece of this whole story. They help to create these pore spaces. They, with this material that is on their bodies, this shiny gel-like material, they actually put this on the surfaces of this pore spaces, and this is a habitat for many microorganisms. So one thing is that they beautifully decompose and aggregate, decompose organic matter and create these amazing aggregates that are called worm castings. That is very important from nutrient availability standpoint, from soil aggregation standpoint. Their bodies are a habitat, their guts are a habitat for beneficial microorganisms, and also they create habitat along the pore spaces, the walls of the pore spaces. They have 5 hearts. That's how much they love everything and everybody. And whenever we degrade the soil and we leave it alone for a little bit, they just come back. For this topic that I covered so far, there is 2 literature that I can recommend for you. Soil Biology Primer is created by the NRCS, the Natural Resource Conservation Service in the United States, and you can freely download a copy of it, and you can also read it online. And there is this book, Teeming with Microbes, which was written actually by journalists, but it's a pretty good work about this whole topic, how to actually work with microbes and how they behave and how all this soil food web behaves. And if you happen to garden, then this is a good handbook that is not too scientific, but helps you to understand how to work with these creatures in your soil. Okay, a little bit more of this. I just gonna have a, oh shoot. I'm going to have a quick look. No. Okay, never mind. Can somebody let me know when we only have 10 more minutes from the class, because I can't see, I don't want to ruin what we have right now, and I can't see the timer very well. Oh, but actually I have a phone here. So it seems like we have half an hour, right? If not so, then please somebody let me know. And then I can also add real quick, if you guys have any questions so far, we have a little bit more material to cover, but from the soil life perspective, I'm happy to answer a few questions if you have.
Hi, Professor. Good morning.
Hello, good morning.
I just have a quick question.
Go for it.
You told us that you do like compost analysis. Are there any like negative microorganisms that you can find?
Oh yeah.
May
Oh
affect
yeah.
like the soil or
Yeah.
um,
Yeah. I can identify some pathogenic fungi and some pathogenic bacteria. So basically, there are certain fungal diseases I haven't brought any pictures of them, but have very specific shapes that is very easy to recognize. Like, for example, Alternaria, Phytophthora, Fusarium. These are pretty easy to recognize by a simple microscope. There is a few bacterial diseases, but actually like pathogenic bacteria that you can recognize by the shape, like for example, spirilla. But those are not mostly plant pathogenic. Those are more human pathogens. That is also a concern from a compost perspective. So yes, and the more you actually train your eye for these, the better you are able to recognize them. But of course, you're never gonna be able to recognize every pathogen because so many of them are actually not visible at the scale that a simple light microscope works with.
okay, yeah. Thank you.
Yeah. And of course, you can also see your efficiency in terms of composting, because if you compost properly, those pathogenic or disease-causing microorganisms die. They are not able to survive proper composting processes.
They die only with the high temperature?
And high temperature and time.
Okay.
For example, if you do vermicomposting, you don't have high temperatures there, but you have, for one thing, a longer composting period. And for a second, one of my mentors says that every pathogenic microorganisms die in a worm's gut.
Okay.
Okay, thank you very much for your question. Anybody else? One more question before we go on.
Yes, Professor.
Okay. Ras phone, right?
Yes.
Go on.
A lot of farmers in my country use like dry manure in our garden. My question is, is there a lot of like bad bacteria or microorganism living in that manure?
What kind of manure is that?
Like cow manure, chicken manure.
Yeah.
Is it good to use it directly? Like,
well, the problem with uncomposted manure is one thing is the presence of pathogens that might be a problem or might not. I believe that you guys have been using these for centuries and nobody's really harmed by them. When you have, so this dry manure, of course, becomes moist in the soil system, so it starts to live again. So many of the microorganisms that were there and became dormant because of the drying process, they become inactive. When they reach the moist soil, they reactivate. And many of these substances that are in the manure, undecomposed organic matter, can be an issue in terms of nutrient imbalances. Just to give you an example, if we are talking about, well, not the cow manure and not the chicken actually, but for example, if we would have dry horse manure, it would be too much carbon and too less nitrogen, and an imbalance would create. And as that organic matter, that carbon decomposes, that would require a lot of nitrogen, so there would be not enough nitrogen for the plants. But that might not be the case with cow manure, because it doesn't have that much intact carbon, like visible plant material. Same thing with the chickens. One thing is that those can burn the plants, you know, because partially because, for example, nitrogen is not stabilized there, as opposed to when you have a compost. Most of the nitrogen is in a stable organic form and just gradually available to plants. So basically, when you apply uncomposted dried manure, then a lot of energy in the soil system is going to be used for the decomposition of that manure, you know, which can be a good thing or a bad thing as well. If you have a living soil, then pathogens, for the most part, should not be a big problem, given that your cows and your chickens are healthy. And this is true more in a small system. Nowadays, it's pretty common to use excessive amounts of dried manure in a pelletized form that is, you know, done in a large scale. That can also cause problems. I'm very into composting, but in a small scale, it cannot really do a huge harm. But it's always better to compost than to dry, you know. And in the drying process, a lot of nutrients leave that material. A lot of available nutrients just go into the air, especially nitrogen. Okay, does that answer your question?
Yes, sir. Thank you.
Okay, all right, so I go on so that we don't we go until 9:45, right? Okay, a silence means yes for me right now. Okay, so this is a lot of things that a soil biota does, a soil microbial population, or not just microbial, but you know, any soil living organism do in the soil, which I already mentioned in this presentation. So this is a summary. They sequester and store carbon, they digest and cycle organic matter, improve the structure of the soil and also the rooting depth for the plants. So they make these nice aggregates and they make the soil accessible for air and accessible for water. They retain, recycle, and solubilize nutrients. So basically, these microorganisms do the nutrient cycling for the most part in a healthy system. They increase water holding capacity. So if a soil biota is present, then with their work, with making aggregates, making more soil organic matter, there is the soil is able to store more water. They are able to decompose toxins. So that's why Rasphone, I told you that actually even when you have a manure that you apply to a soil, but your soil is alive, then it's able to deal with those potential pathogens. So a few pathogens is not going to harm a healthy soil. Okay, it's always a problem in an unhealthy soil where there is not enough microorganisms to decompose the toxins or kill, eat alive the pathogenic microorganisms. And that's the second point, they are able to suppress disease. But also, like the decomposed toxin stories, that I'm working with regenerative farmers, and they are conventional farmers, so they use chemicals, but they do every possible measure that I will talk about later to increase soil health, to increase soil life. So there is no traces of the chemicals that they use. They use herbicides, but these herbicide residues get decomposed in their soil system very quickly because they have a very lively soil. They also use some of them. At some point, they stop using these as their soils become healthier, but some of them uses fungicides or other insect killer chemicals, and it's not able to harm their systems very much because their soils are alive, and I will show you how they make their soils alive. And again, yeah. Yeah, go for it.
I'm pretty sure you know about the effective microorganism. In my area, people also use the effective microorganism, or EM.
Yeah.
That is condensed in the liquid form, and then they spray that into the manure or the soil before using them and leaving them for maybe a while, a week, so that the EM colonize, maybe eat or destroy the bad pathogens.
Yeah.
Is that also possible? Like, because you are...
absolutely.
Can you still hear me? I have bad connection quality.
Say your last sentence again.
Did you lose me?
I didn't understand your last sentence. Can you repeat?
Because of we spray a lot of microorganisms, so somehow I think those microorganisms also neutralize the bad pathogen as well, right?
Yeah, that's absolutely true. And there is more different schools, to do this. There is those who believe in AM, and there is those who believe in compost-based products. And I don't know enough about AM. I have actually never tried to give you a very well-educated pro or contra story. But what I grasp of this is that actually it's mostly bacilli, facultative anaerobic microorganisms, because these are quite essentially fermented in water, or brewed in water. So this is mostly lactobacilli. And there is very many studies for mostly for that's what actually I have been looking at, non-aerated compostes, which have probably very similar microbial composition as the AM. They are very good at suppressing diseases. But I believe that actually any compost extract, like for example, if you make which is much cheaper, a watery extract of composts, simply just shake your compost with water and then filter it and spray that onto your soil or onto your plants. It has this disease suppressing effect. I have done experiments with this where I have been sprouting mustard seeds, and those seeds that I treated with compost extract with a good chunk of compost microorganisms, they grew beautifully. And those that I have not treated with compost, only with DI water, those became moldy. But there is much more sophisticated studies about it. So if you are interested in this, just feel free to search the scientific literature, because there is plethora of information about it. Yeah. Okay. Thank you for your question.
Thank you.
All right, let's go on a little bit. So basically, how can we support the soil ecosystem? How can we make sure that these creatures live and thrive in our soils? There is a set of principles which is called the soil health principles that I'm going to show you real quick now, which is minimize disturbance. I mentioned so many times that tillage disrupts this entire system, and we have to make sure that we find other methods to make a habitable place for our plants, and and tillage is not a solution for this, but it's actually what causes the problem. For the most part, it's tillage what causes our problems. We always have to cover the soil. We have to have roots 24/7. There must be always roots in the soil because that is the habitat for microorganisms. We need to increase diversity, and also we need to integrate animals. And I'm going to break these out and give a few examples. So minimize disturbance. There is a regenerative farmer movement, essentially, where some farmers, quite a few farmers now actually, just simply stopped using the plow and turning around the soil. And many of them don't even scratch the surface, just directly seed their next crop into the previous crop's residue. I'm not going to get into this now. Basically, it's very important that long-term non-tillage increases soil microbial community, increases their diversity, and also increases their stability. So if we stop plowing, we will have more and more diverse microbial population. Okay. So the second thing is always cover. So which means that we have to have something on the soil surface. You know, when the rain is falling or the wind is blowing, that is very destructive to the soil. And soil life, most of the soil microorganisms are on the top 10 centimeter of the soil. So if they are exposed to severe weather or just simply to the energy of the sunshine, that is very destructive to them. That's one reason why we have to have cover, to protect it from physical, to protect the soil surface from physical effects. The second thing is that with this cover, we also, if it's a living cover and an organic cover, then we have an organic matter replenishment for the soil. And that adds to the have living roots 24/7. Oh, this is the cover still, which keeps the soil together. And I'm showing you here an experiment. This is very interesting. It's a rainwater simulator. So basically what they do, they take intact soil samples and put them into these trays. Okay, and the first one is a disturbed soil, doesn't have any surface coverage. The second one has a little bit of surface coverage. The middle one has quite a lot of cover, and the fourth one still more cover, and the last one has a living cover. So basically, when they make a rainfall, every single box gets the exact same amount of rain. Okay, and when it rains, you can see that the surface runoff is being collected in front of this rainwater simulator. And when the rain hits the bare soil, then you get a lot of runoff. Water is not able to infiltrate into the soil. It's running off the surface. And you see that the water brings a lot of sediment with it. This is how we lose our agricultural soils. If you have just a tiny little bit of cover, there is much less water being lost and more can infiltrate. That little bit of organic matter can stop the water flow or slow the water flow and let the water infiltrate into the soil. Water is life, and all microorganisms and everybody needs to have water to be able to live. Again, you have more and more cover, the less and less surface runoff you have. And if you have a living cover, there is no runoff. That living cover is able to keep that water in there and lets the water infiltrate into the soil along the root channels, and also just simply by keeping the water for a longer period of time on a given surface. And now here in the bottom, you see how much water is able to infiltrate into the soil. So if there is no cover, then no water gets into the below soil parts. After like maybe 1 or 2 centimeters is able to get wet, and that is, and the water is not doesn't penetrate deeper down. Again, here you have a little bit of water infiltrated, and the more cover you have, the more water is being infiltrated. But as you see, if you don't have living roots, the water is washing down fine particles with it that accumulates in a deeper surface layers, which can cause a compaction problem. It can cause a nutrient availability problem, and so on. But if you have a good surface cover, a living surface cover, you have clean water in the bottom. How simple is that? Okay, we have 10 more minutes. This is the Rastafarian roots that I mentioned. And these are these rhizosheets, the mineral particles, along with a lot of organic matter, a lot of microorganisms, the microbial glues, the polysaccharides from the bacteria, the glomalin from the fungi that stick all these together. And this is where all this nutrient cycling happens. And this is what feeds the plants. This is something that I already talked about, the image of these rhizosheets, all the bacteria, the root cap, the root exudates, the mycorrhizal fungi, and so on. This is the rhizophagy cycle that is very important in terms of roots. This is very simply, maybe I have an image. Bacteria not only help nutrient uptake in the rhizosphere, but actually they are able to enter the root hairs through the root tip, and then they give up their nutrients inside the plant. And then, actually it's at the root tip, and then through the root hairs, these bacteria leave and go and eat again, take up some nutrients and go back. This is the rhizophagy cycle, where bacteria actually enters the plant through the root and feeds the plant directly inside the root system. Isn't that amazing? Okay, increased diversity, very important, because most agricultural pests like to have monocultures. It's easy because there is one plant that they like and there is a lot of them, and they just go ahead and have a feast and increase in numbers and have a good time. But if you have a diverse system, they just get lost. That's one of the reasons. And the other, if you have certain type of plants, like for example dill or fennel, that is also attracts predatory insects, like for example certain wasps that feed on pathogenic organisms. So diversity is very important in these systems. And there was a study, this was a meta-analysis where they actually looked at how agricultural diversification gave a lot of ecosystem services without actually losing yield. So you have the same amount of corn, but you have a lot of ecosystem services. Like for example, soil fertility, water regulation, pest control, nutrient cycling. This is just an image from the same article that you actually see how diversity promotes soil fertility, how it affects crop yield, and so on. Let's not get into this, so we have a few more minutes for questions. Integrate animals. So basically, regenerative farmers plant cover crops between the growing seasons, which means that they always have living roots, they always have cover, and they have also nutrients being captured and held at place by those cover crops. Cover crops can be different type of legumes or different type of grassy plants, cereal plants, and so on and so forth. And many farmers actually like to, more and more farmers, unfortunately not too many, but more and more farmers like to graze these cover crops. So they have an additional benefit of being able to feed their cattle herd, for example, from cover crops. So they graze the cover crops before planting the next cash crop. Okay, so some recommended readings if you are interested in these topics. Nicole Masters, For the Love of Soil. She's a soil life consultant. She's really a brilliant woman, and this is a pretty nice book. You also find a lot of lectures that she gives online. And then Gabe Brown, he's probably, I don't really like the word guru, but really he's the guru of regenerative agriculture. He's one of the first ones out there who started more than 40 years ago the journey into regenerative agriculture. And his story is really, really nice, and you can learn a lot from him. And then the last one is a textbook. This is called The Fundamentals of Soil Ecology. And it's a really nice book. This is actually one of my favorite soil ecology books. So if you want to dive deeper into the science of soil ecology, then I would probably recommend you this book. Okay, so I think this was it. And I really appreciate your attention and your few questions. And I think we have another 4 or 5 minutes for questions if you have any, or any feedbacks that you would like to provide. So feel free.
Hello.
For
me, I don't have any question. Thank you so much for your kind presentation.
You are very welcome. My pleasure.
Hello. Yeah, also for me, everything was clear. Thank you very much. I was just wondering if we can get like the slides, because they are very interesting, like to get also the articles, to see where the articles are on.
Okay, sure.
Please.
I will ask Professor Kardosh to upload it to e-learning, okay?
Okay.
And if that, maybe actually, because I have a lot of videos and everything, I will see how big the file is. If it's too big, then we might end up sharing it through Google Drive or something, okay?
Okay. Thank you very much. It was very interesting.
Okay, you are welcome.
Bye-bye.
Okay, bye. If nobody has any more questions, then you guys have a wonderful rest of the week and rest of the day, and have fun studying.
Thank you.